Hello, this is Jasmine Trigg, and I'm a scientist at Sartorius. Today I'll be discussing a poster that I previously presented at AACR, and this is titled Multifaceted Characterization of Ferroptosis using Live-Cell Imaging and AI-Driven Analysis. So I'll start with a quick introduction. Ferroptosis, a form of regulated cell death, is distinct from apoptosis, and this represents a new mechanism by which cancer cells can be targeted and killed. Here we used live cell imaging to capture the dynamic morphological changes that are associated with ferroptosis and to characterize this mechanism cell death, as well as look at it in contrast to apoptosis. We utilized the Incucyte AI cell health analysis software module, and this is driven by pre-trained neural networks and is a robust solution for label-free segmentation and live-dead classification of ferroptotic cells. Additionally, we use the Incucyte advanced label-free classification software to perform machine learning-based classification of cells in early stages of ferroptosis based on morphology. So the data that I'm going to show you has been acquired using the Incucyte Live-Cell Analysis System, and this is an integrated solution of hardware, software, reagents, and applications that are all designed to work together. So the hardware itself is a microscope optimized to permanently reside inside a standard lab incubator, and then we have objectives that move around underneath the culture flasks or microplates and acquire phase and fluorescence images at regular intervals. We can also connect remotely to the Incucyte from any location, and this allows us to kind of remotely schedule scans or look at the data in real time. Alongside this, we provide a suite of non-perturbing reagents and consumables, and this allows you to run the assays. And we have a number of applications across a number of different research areas of biology. So if we look at the assay principle here, so ferroptosis occurs because of a redox imbalance within the cell. And here we see that there's an increase in intracellular iron, and this drives lipid paradoxation, and this is regulated by GPX4. Inhibitors of this process, such as RSL3, FIM56, or Erastin, cause lipid-reactive species to accumulate, and this then induces ferroptosis. So we've got kind of an example schematic on the left here, which shows the expected morphology of ferroptotic cells, and this is distinct from that of both apoptotic and necrotic cells. On the right here, we've got some example Incucyte images, and this is of A549 cells that are expressing a nuclear restricted fluorescence protein in the NIR channel, and these are treated with FIM56. And if you look at the images over time, we can see that there's a temporal change in morphology as the cells undergo ferroptosis. So firstly, we'll start by looking at AI-driven label-free quantification of ferroptosis. So here, HeLa cells are treated with concentration ranges of the apoptosis inducer CMP and ferroptosis inducers Erastin, FIM56, and RSL3. We then performed AI cell health label-free analysis to segment the cells and classify them as live or dead. So if we look at the kind of the left half of the figure, this here has some representative phase images, and these show that the vehicle cells were classified as live, as you can see by the kind of the green segmentation, and that both camptothecin and FIM56 treated cells were classed as dead, as you can see with the red outline there. If we look at the histogram, this displays the predicted dead scores for the live cells, apoptotic and ferroptotic cell populations. And you can see that we've got clear separation between the live and dead populations, with ferroptotic and apoptotic cells showing a similar range of dead scores. This indicates that ARC Health can accurately classify ferroptotic cells as dead and provide a label-free readout for total cell death. If we look at the microplate here on the right, this shows the percentage of dead cells over time, and we can see that all compounds that we tested induced a kind of a time and a concentration dependent increase in cell death compared to vehicle. So here we're looking at the pharmacological inhibition of ferroptosis. So HeLa cells were treated with camptothecin and auristatin in the presence or absence of the of ferrostatin 1, which is an inhibitor of ferroptosis, or ZVAD-FMK, which is an inhibitor of apoptosis. And this is a really nice validation tool to kind of confirm that we are inducing apoptosis when we think we are, as well as inducing ferroptosis when we think we are. So if we look at the time courses in A, this shows the percentage of dead cells in response to either camptothecin or auristatin, with or without ferrostatin 1. So you can see that ferrostatin 1 had no impact on apoptosis induced by camptothecin, and then you can, in contrast, we see that auristatin alone induced cell death up to 60%, and this was reduced in the presence of ferrostatin 1. Conversely, if we look at B, we can see that when ZVAD was included with auristatin, there was no reduced cell death, but the concentration response curves confirm that auristatin cell death is inhibited by ferrostatin and not by ZVAD. So the next two graphs are looking at the density dependency here. So HeLa cells at 4 seeded densities were treated with auristatin or the apoptosis inducer staurosporin, and the concentration responses show differences in cell death in the presence of auristatin but not in the presence of staurosporin. And this is in line with ferroptosis being reduced in highly dense cultures with a number of cell-to-cell contacts. So here we're looking at how we can identify early ferroptosis label-free. So HeLa cells were treated with the ferroptosis inducer RSL3. These were then segmented using cell by cell, and then label-free classification was used to identify morphologically distinct transition state of the early ferroptosis and cell death. So you've got a representative image on the left here at 6 hours post-treatment, and this shows that label-free segmentation and classification of the healthy cells shown in teal and the early ferroptosis cells in magenta. And this shows how we can really kind of identify that early morphology. So if we look at the time course, this shows the percentage change in early ferroptosis and cell death for RSL3 compared to vehicle. So for cell death, we see this rapid increase over 12 hours, and we can see that ferroptotic cells show this earlier peak as the cells pass through that transition state prior to cell death. The label-free classification of early ferroptosis was then applied to a small compound screen, and this included 24 compounds at high and low concentrations. So if we look at the bar chart on the right here, this displays early ferroptotic cells for high concentrations. And we can see that the early ferroptosis phenotype was only present for ferroptosis inhibitors, which is good, it's kind of aligned with what we want. And we also saw this with Taxol, and interestingly, this is a microtubule stabilizing agent, and this has a lot of shared mechanisms of cell death to ferroptosis. So the next two figures are going to look at how we can quantify ferroptosis using fluorescence readouts. So in this example on the left here, we had normal human dermal fibroblasts, and these were treated with FIN56, and then the loss of membrane integrity was assessed using the Incucyte cytotox green dye. So we can see we've got some images shown here at 36 hours for either vehicle or FIN56, and we can see that alongside this and the time course, that FIN56 induced rapid and concentration-dependent cell death over time compared to vehicle. In the example on the right, here we're using non-adherent Ramos cells. So these cells were stably expressing a nuclear-restricted fluorescence protein, which is the nut-like orange, and these were then treated with auristatin, FIN56, or RSL3. We then used non-adherent cell-by-cell analysis to quantify the mean intensity within the cell population, and we've got representative images here at 48 hours. The curves confirm that all of the compounds induce cell death in a concentration-dependent manner, and you can see we've got slight differentials in potency across the concentration ranges and compounds tested. So here we're looking at ferroptosis in a 3D single spheroid model. So A549 cells expressing the Incucyte Nuclei NAR were seeded into a ultra-low attachment round bottom plate, and these are then allowed to form for 4 days. The spheroids are then treated with camptothecin to induce apoptosis or erastin to induce ferroptosis, and spheroid bright field area and fluorescence intensity was quantified. So we've got some representative images here of either the NER or nuclear fluorescence with the segmentation mask, as well as the bright field images. So we can see that the quantification of the nuclear intensity show that camptothecin and erastin induce cell death in spheroids, and we have a decrease in the nuclear fluorescence as they die. The bright field images revealed that treatments induced different morphologies. So camptothecin causes shrinkage and the spheroids remain very dark and compact. In contrast, erastin causes loss of compactness. And if we look at the time courses quantifying bright field area, this confirms this. So we see the decrease in area for camptothecin and a rapid increase in area for erastin. So in summary, live cell imaging enables acquisition of high-definition phase contrast images and provides an ideal platform to study multifaceted biological paradigms in drug discovery. Here we've quantified ferroptosis in a range of 2D and 3D models using label-free AI-driven analyses or fluorescence reporters of cell death. Overall, the data exemplify that the Incucyte, alongside advanced AI-driven analytics, is a valuable platform for characterizing the dynamic process of ferroptotic cell death. And this really yields insights into the cellular mechanisms and enables quantification across a range of research models and multiple cell types. Thank you for listening.